Biochemistry 401, Lecture 22. Today we're going to talk about ATP synthesis, uncouplers of oxidative phosphorylation, and the poisons that affect oxidative phosphorylation. Oxidative phosphorylation consists of processes that are carried out by the electron transport chain, which we saw in the last lecture, and also ATP synthase. These processes of oxidative phosphorylation consume oxygen and synthesize ATP. We know that the electron transport chain uses the delta G of electron transfer in order to pump protons from the mitochondrial matrix into the intermembrane space. This forms a chemiosmotic gradient. So what is this chemiosmotic gradient? It's the proton concentration gradient. But because protons are positively charged, it's also an electrical potential across the inner membrane. And in this way, the difference in the concentration of protons sets up a charge difference and an osmotic difference that causes protons to seek to flow from the intermembrane space back into the mitochondrial matrix. And so the proton motive force, delta P, is equal to the chemical gradient plus a charge gradient. And so protons seek to flow through the inner membrane back into the matrix. But how do they do that? Well, one of the ways that protons can travel across the inner mitochondrial membrane is to flow through ATP synthase. This causes the turbine-like motor in ATP synthase to turn. And this results in conformational changes in ATP synthase that allows the production of ATP to happen. Let's take a look at this remarkable enzyme, ATP synthase. You will recall that the inner mitochondrial membrane is thrown into a series of folds that are called cristae. ATP synthase is found in double rows at the ends of these cristae, and it is thought that ATP synthase dimers actually help to form cristae and maintain their structure. Now that we have more sophisticated tools, we can look at enzymes on an individual basis. For instance, this image was created by cryo-EM, which involves freezing a sample and then looking at it with an electron microscope. This micrograph has been pseudo-colored to show individual subunits that comprise this ATP synthase complex. A ribbon diagram derived from X-ray crystallographic structure of ATP synthase has been superimposed onto this cryo-EM micrograph to show the placement of individual subunits. Let's take a look at the individual subunits that are found in ATP synthase. Unfortunately, there's a discrepancy in the naming of subunits between bacteria, which is a prokaryote, and eukaryotes. This diagram shows bacterial structures on the left and eukaryote yeast on the right. In this lecture, we're going to be using the naming that's seen in eukaryotes. And in this diagram, the matrix is shown at the top of the slide and the intermembrane space is shown at the bottom. In both bacteria and in yeast, these subunits that are found in ATP synthase are grouped into two main structures, the F0 and the F1. The F0 is found in the inner mitochondrial membrane, and F1 projects into the mitochondrial matrix. F1 contains the subunits that are going to be directly involved in the production of ATP synthesis, whereas subunits in F0 provide the rotation that allows ATP synthesis to happen. In order for ATP synthase to function, some components are going to rotate while others remain stationary. The rotating components include the C-ring and a central stalk that's comprised of gamma, epsilon, and delta structures. The C-ring starts the rotation, and because of the association of the central stalk with the C-ring, the central stalk also rotates. The stationary components contribute to the stator and also the hexameric ring, comprised of alpha-3, beta-3 subunits. The production of ATP from ADP and inorganic phosphate actually occurs in the hexameric ring, comprised of alpha-3 and beta-3 subunits. So what do you need to know? You need to know what the alpha-3, beta-3 hexameric ring is, and the central stalk, the C-ring, the alpha subunit, and the stator. And of these components, you must know which move and which don't. So again, the C ring rotates, the central stalk rotates, but because of the stator that's firmly anchored in the inner mitochondrial membrane, the alpha 3 beta 3 hexameric ring does not rotate. And this is important to know. But how is it that the C ring moves? What provides that momentum? Well, this has to do with the way protons enter the mitochondrial matrix from the intermembrane space. We said that these protons just can't go right through the membrane. They have to have a way through. And this way through is through subunit A. But the thing is, the protons just don't go right straight through subunit A. Subunit A only has two half channels, one that begins in the intramembrane space and the other one that ends in the matrix. But what about this middle part? Where do the protons go then? Once they enter from the intramembrane space, how do they get to the matrix if this is only a half channel? The solution lies in the C ring. The C ring is made up of several C subunits arranged in a ring. Each subunit has a helix, loop, helix structure with an important aspartic acid in one of those helices. Let's see how this aspartic acid contributes to the rotation of the C ring. In this figure, we have three panels, A, B, and C. In the A panel, we have the C ring and subunit A, shown from the side and also from the top. Subunit A is shown in cyan, and the C ring is shown in brown. In panel B, we see the amino acid aspartate, and this is the conjugate base of aspartic acid. The R group of aspartate can be protonated to form aspartic acid, and this is how protons get from one place to the other. They come in through the intermembrane half channel, they protonate aspartate to make aspartic acid, then the ring rotates and drops off the proton at the other half channel so that the amino acid becomes aspartate again, and the proton leaves to enter the matrix. Let's look at this again bit by bit. I like to think of this in terms of a carousel, with protons being the brass ring. As the children come over to the place where the protons enter, they grab the proton, the merry-go-round revolves around to the other side, and the proton is released in the other half channel to enter the matrix. Now, why does this happen? We know that aspartate is negatively charged, and as such, it's going to have a hard time entering the plane of the inner mitochondrial membrane. However, when it's protonated to form aspartic acid, now this R group has a neutral charge, so there's no problem with the aspartic acid going into the plane of the membrane. And that's what it does until it hits the proton-poor environment of the matrix half channel. And then aspartic acid becomes deprotonated again to form aspartate. The child tosses the brass ring away and reaches out to grab a new one. The video that we see below was derived from cryo-EM micrographs and shows the rotation of a C ring. As the C ring rotates, because of the close association between the C ring and the central stalk will also rotate. And as you can see, the gamma subunit of the central stalk, also known as a shaft, is asymmetric. And so when this central stalk rotates within the hexameric ring, it's going to bump into the ring and cause conformational changes in the subunits of the hexameric ring. Now, the hexameric ring is made up of alpha and beta subunits that alternate around the ring. The beta subunits are the ones that are catalytic, and the alpha subunits are structural to keep the ring together. Now, as the central stalk bumps into these beta subunits, it's going to cause conformational changes in that subunit, depending on what portion of the stalk is actually contacting that subunit. Let's take a closer look at what's actually going on. In this figure, we're looking at the alpha 3 beta 3 hexameric ring, and the beta subunits are colored in purple, lavender, and cyan. We're going to look at just the purple colored beta subunit and see what happens to it as the central stalk bumps into it. The three major conformational changes that happen are open conformation, loose conformation, and tense conformation. In the open conformation, which is the one we see in number 1 and 4, there's a nucleotide exchange that happens. ATP leaves and is replaced by ADP, an inorganic phosphate. Now, as the stalk rotates 120 degrees counterclockwise, the purple subunit becomes in the loose conformation, and this is more or less a holding pattern. Nothing much happens. But then with another 120 degree rotation, the purple subunit is going to be in the tense conformation. And in this tense conformation, the subunit will be in such a shape that it becomes easier for ATP to be synthesized from the ADP and inorganic phosphate. And so ATP is formed. Now with one more 120 degree rotation, this purple subunit opens again, releases the ATP that was just made, and takes in more substrates, ADP and inorganic phosphate, to start the cycle again. And here is a video that's showing the counterclockwise rotation of the central stalk and the conformational changes in the beta subunits that allow ATP to be synthesized and released. Now that's one way that protons get from the intermembrane space to the matrix. But there are other ways, and one of them is by way of a protein called thermogenin or uncoupler 1. This is a protein that inserts into the inner membrane that allows protons to be transported through this inner membrane and into the matrix. Now the thing is, when this happens, we're releasing some of the potential energy of that chemiosmotic gradient. And so when that energy is released, something has to happen. Remember, energy is just changed. It's never lost. And so what happens? This potential energy is changed in part to heat and in part to entropy. And so this thermogenin, as the name implies, makes heat. And this is a way that animals can get warm. This is called non-shivering thermogenesis. Thermogenin is a protein that's found in brown fat, and brown fat is useful in maintaining body temperature. It's highly represented in infants, and as we get older, we have less and less brown adipose tissue. But it's still there. And here is an actual micrograph of brown fat tissue. And next to it is a study that was done with men who were thin and men who were overweight, and looked at the location and the activity of brown fat in these individuals. And it's found that when the temperature of the room was lowered so that these men were cold, the activity in brown fat increased. And there was a more substantial increase in men who were lean than there was in men who were overweight. And why is this protein called uncoupler 1? This is because we're uncoupling the electron transport chain from ATP synthesis. So we're uncoupling the proton gradient from the production of ATP. Another thing that's an uncoupler is called 2,4-dinitrophenol, and this is something that's used in the production of TNT that's found in dynamite. And this is also an uncoupler. And so persons who were working in munitions would have symptoms in which they would lose weight and they would have fevers, and nobody knew what was happening, but they knew that this was something that was happening to these people who were working in these factories. And this is because the DMP was binding protons and carrying them through the inner mitochondrial membrane and into the matrix. And so again, we have the production of heat. The scary thing about this, though, is that this is also packaged as a diet aid. And recently there was a woman in England who took this as a diet aid. And she took 8 capsules. Don't know why she did that, but she went to the hospital burning with fever, and there was nothing that they could do for her. She literally burned from the inside, and she died. Her temperature skyrocketed. There was nothing that they could do because this DMP was just allowing the proton gradient to flow right through that membrane, allowing the potential energy to be turned into heat. And this is a vicious cycle, right? The body thinks it doesn't have enough ATP, and so it ramps up those catabolic pathways that would produce ATP, and the proton motive force is regenerated, and as soon as it's regenerated, it's lost to heat again. And so it's a vicious cycle, and literally this woman died from uncontrolled fever. Really scary stuff. And so thermogenin and DNP are both uncouplers of oxidative phosphorylation. These uncouple the oxidative part from the phosphorylation part. So there are other things that can poison this process as well, and these include things that poison the electron transport chain and things that poison ATP synthase itself. These are rotenone and amytal, which poison complex 1, antimycin, which poisons complex 3, cyanide, azide, hydrogen sulfide, and carbon monoxide, which poison complex 4, and then oligomycin, which poisons ATP synthase. So say, for instance, a person goes down into a manure pit for some problem and is overcome by hydrogen sulfide gas. What's happening is cytochrome C oxidase, complex 4, is not able to function. What this means is electron transport complexes before this are going to remain in the reduced state, and oxygen is no longer going to be consumed, ATP is no longer going to be made, and the body cannot function without ATP. Now say, for instance, ATP synthase was poisoned by oligomycin. This means initially the electron transport chain is going to be functioning just fine, pumping protons into the intramembrane space. But if those protons can't get out of the intramembrane space, the gradient is going to become so large that the electron transporters are not going to be able to pump protons against that gradient any longer. And so eventually the electron transport chain is going to slow and stop because the concentration gradient is much too great for these proton pumps to pump against it. So say, for instance, though, you did an experiment in which you poisoned mitochondria with oligomycin to block ATP synthesis, and eventually the electron transport chain slowed down and stopped. But then you treated these mitochondria with the uncoupler DNP. What would happen? Well, the electron transport chain would get started again once the gradient became sufficiently low for this process to keep going, and oxygen would be consumed. But ATP synthesis would still not happen, and so you would have the electron transport chain running, but no ATP being produced. So how much ATP can you get from NADH? How much can you get from FADH2? To understand how much ATP can be produced, you'd have to take these electron carriers, NADH and FADH2, one at a time. So for instance, let's say we start with electrons that come in by way of NADH. These are going to go through the electron transport chain and eventually reduce molecular oxygen to produce water. For every NADH that enters, 2.5 molecules of ATP are generated. Likewise, for every FADH2 that enters and reduces molecular oxygen, 1.5 ATPs are going to be generated. Why is there a difference in these two values? Well, don't forget, NADH enters by way of complex 1, and so it generates more of a proton gradient because complex 1 can pump protons. Electrons that come in by way of FADH2 enter through complex 2 or through ubiquinone, and so these completely bypass complex 1 and only generate the proton gradients by way of complexes 3 and 4. And so since there's less of a proton gradient generated by FADH2, less ATP are going to be generated from these electrons, and that's it. You're going to have to be able to calculate how much ATP can be generated from a mole of glucose or from a mole of pyruvate. And so these are basic calculations that you can make, figuring out how much ATP are produced directly from those carbon sources, how much NADH and how much FADH2 are created from the oxidation of those carbon fuels. Then multiply by the correct PO factor and add them up. And you're going to have to be able to do this for the next exam. And this marks the end of Lecture 22. I hope you enjoyed it. Have a great day.